Okay, so lymphatic system is today. Again, super straightforward. We're going to list the functions of the lymphatic vessels, talk about their structure and their distribution. We'll talk about the source of lymph and how we transport it. We'll talk about the basic structure and cell population of lymphoid tissue. We'll talk about the location, histological structure, meaning the tissue structure and functions of lymph nodes. We'll compare and contrast the structure and function of spleen in the lymph nodes. We'll talk about something called MALT, which is mucosa-associated lymphoid tissue and its major components. And then we'll talk about the thymus, which you guys are all already familiar with. All right, so the first key concept is that the lymphatic system includes lymphatic vessels, lymph, which is the fluid, and lymph nodes. And so we learned all about capillary dynamics with blood vessels. And so one of the functions of the lymphatic system that you should already know is that it picks up excess interstitial fluid and leaks proteins at the capillaries. So when we have capillary exchange, we have fluid along with oxygen and nutrients going out on the arterial end. Most of it gets sucked back up along with waste products on the venous end, but then there's still fluid left over that your lymphatic capillaries pick up. So they pick up the excess fluid that your capillaries cannot pick up. And so a quick review from B32 of polymers and monomers. The polymer is a nucleic acid, but what is the monomer for a nucleic acid? Nucleotide. What about for carbohydrates? Monosaccharides. For protein, amino acid. And although lipids don't have a true monomer, what do they break down into? Do you know? Fatty acids and glycerol. Good. Now the lacteals, which are a specific or a very specialized capillary for the lymphatic system, their job is to pick up lipids specifically. So when we bring in new foods, when we eat new foods, the capillaries at our intestines can pick up the nucleotides, they can pick up the amino acids, they can pick up the monosaccharides, but they cannot pick up fat. And so we have these specialized capillaries called lacteals, where their job is to absorb the fats from the food that we eat. Those fats will eventually go into our bloodstream, but they have to take a detour through the lymphatic system first. So that brings us to our next function of the lymphatic system, is that lacteals absorb fat, which actually gets converted into a fluid called chyle. Okay, so chyle essentially is it's lymph, which is our lymphatic fluid, plus fat. And it's white. It's a milky white lymph because it is fatty. So what you're seeing on this image here are intestinal villi. You should have learned about villi and microvilli in Bio 32. Does anybody remember what this tissue type is that lines our intestines? Simple columnar epithelium. So within these villi of the intestines, there are these green capillaries or vessels that you see right here in the middle. So this green vessel here, that is a lacteal. And so a lacteal, we can define that as a specialized lymphatic capillary in the small intestines specifically. Anybody remember what the function of villi are? What do they do for us? They increase surface area, yes. Yay! So make sure that you know that villi increase surface area. makes it so that we can absorb the maximum amount of nutrients from our food, because if our intestines were just flat, that has a lot less surface area to absorb food from than with the villi. Villi just give us so much area to absorb more nutrients, and so absorption is just a lot more efficient with those villi. More area for more absorption. Okay. Questions about lacteals. They absorb fat. When it mixes with that lymph that's inside that capillary, it becomes something called chyle. Okay, so make sure that you remember that chyle. We're going to learn a new term after respiratory when we get to digestive. The fluid that's in your stomach is called chyme with an M. So you're going to have to make sure that you get those two fluids straight. Chyle has an L, so it's lymphatic. And then chyme has an M, like for food. All right, so function number 3 is that the lymphatic system, its job is to defend and protect us from invaders. So one thing I want you to remember, and I want you to highlight it, circle it, put a star next to it. The main job of our lymphocytes, because our lymphocytes are our main cell type for the lymphatic system. Our second cell type is monocytes. But we see lymphocytes most often. Those are our major cell type of the lymphatic system. Their job is to survey fluid. So we'll talk about lymph nodes here in a second. Everyone say happy belated birthday to Savannah as well. Her birthday was yesterday too. Happy birthday. So the lymphocytes, their job is to survey the fluid. What you're seeing here, and what we'll talk about later, is a lymph node. Lymphocytes live inside this lymph node here. And so as fluid is going through, their main job is just to survey the fluid. Okay? If there's something that doesn't belong, then they'll capture it, they'll destroy it. But their main job, what they're doing most of the time, is just surveying the fluid, looking for anything that doesn't belong. So on an exam, hint, hint, if I ask you what the main function of the cells of the lymphatic system is, their main job is to survey, or surveillance. That is their main job. Okay? Only when they find something do they do something about it. But their main job is just to survey the fluid. Yeah?
Is that for the lymphocyte and the monocyte?
For both, yes. If I were to ask you what the main cell type for the lymphatic system is, it's lymphocytes first, monocytes are secondary.
They're both surveying.
They're both surveying fluid, yes. All right, so for the distribution and structure of lymphatic vessels, we have different components we need to talk about. So we have the lymph fluid, which is the fluid that's being transported through the system. We have lymph capillaries, which are the ones that are absorbing that fluid at our arterial and venous capillary beds. Then we have lymph vessels, which those capillaries are going to drain into. And then eventually we drain that fluid back into our blood system, back into circulation. So we're just picking up the excess fluid at the capillaries, transporting them through lymph nodes all the way back till we get to the veins. So we're actually going to dump that lymph fluid into our subclavian veins, and then they return back into circulation. So first is our lymph, which is essentially excess interstitial fluid. These lymph capillaries are just picking up the excess fluid that the capillary bed cannot pick up itself. It is basically plasma with much fewer proteins. If you remember when we talked about those 3 fluid compartments, we have lots of protein inside of our cells because we have ribosomes to make them. We have lots of protein inside of our blood because our liver makes proteins to put into the blood. That interstitial fluid, that middle space though, should have very little to no protein in there. Okay? So that fluid you see in the lymph system is basically plasma, but not as many proteins. Okay, little protein. This is the same fluid that you would see inside of a blister. And it is sterile fluid, so it's always important not to pop a blister because then you expose the inside of your body to potentially bacteria, viruses, things like that. Okay, so next are the lymphatic capillaries. How they work is they have these flap-like mini valves that increase permeability. So this allows proteins, pathogens, which pathogens are disease-causing organisms. They're usually microorganisms. But it allows proteins, pathogens, etc., to easily enter the lymphatic system. So how lymphatic capillaries work is that these valves open and whatever fits through will go through. Okay, there's like with our regular capillaries, we have like continuous capillaries, we have fenestrated capillaries, we have sinusoid capillaries that kind of determine how permeable those capillaries are. Here, they open and anything is let through. Okay, so they're very permeable. And how they work and how they function is that they open under interstitial pressure. So what that means is, I'm going to make a drawing to show you how this works. Okay, so we have these flaps here. So this is a lymphatic capillary, and we'll have lymph inside of here. So at the capillary bed where gases and nutrients and stuff are being exchanged and where the lymphatic capillaries have to pick up that excess fluid, there's going to be excess fluid all around this lymphatic capillary, which is going to increase the pressure around that capillary. Once it experiences that increase in pressure, then what's going to happen is that these flaps are going to open. Ooh, go back. No! Okay, well you have the one with the fluid around it, so I'll draw this one again. So once we have the fluid around, these flaps are going to open and allow fluid to come inside, just from the pressure of the fluid on the outside. Okay? So then, once the fluid comes inside, it's then going to build pressure from within. So fluid inside is pushing up against the wall of the capillary from the inside, and that's going to make them close again. Okay. So it builds up pressure on the outside, flaps open, fluid comes inside. Once the pressure builds up on the inside of the capillary, then those valves close again. Okay. Does that make sense? Okay. Any questions about how that works? So they're opening and closing under that interstitial pressure. Okay. And all that is happening at our cardiovascular capillary beds, and they are picking up that excess fluid.
You said that once there's excess fluid inside the
Capillary, the valves will close. Yeah, yeah. So they open from outside pressure, interstitial pressure, and then they'll close from the inside pressure of the capillary once it fills up with enough fluid. So lymphatic vessels, it's just a unidirectional flow toward the heart. We pick up the excess fluid at the capillary beds, and then we dump them into the subclavians. No matter where it's coming from, it's going to the subclavians on either side. Larger vessels are formed from smaller ones. So we start with lymphatic capillaries, and the vessels get bigger and bigger and bigger all the way until we approach the subclavian veins. So the order of these vessels from smallest to largest are lymphatic capillary, those will then form what are called collecting vessels. Those collecting vessels will then form lymphatic trunks. And those trunks are going to form ducts, lymphatic ducts. And this is not like blood vessels. You're not going to have to memorize every single name of every single lymphatic capillary or vessel. Just know the order in general, from capillaries to vessels to trunks, then to ducts. Now, the more complicated part of this is how they're drained, because the drainage of our lymphatic fluid is asymmetrical. So it's not drained evenly on either side. Don't know why our bodies are like this. So you see how there's a green section in the top right-hand corner of his body, and then the rest is skin colored. That's how the drainage goes. So the left side of the body, so your lower limbs and your intestines and your lumbar trunk, so on the back of your body, all of that fluid, so from your legs and your abdomen and your back, all of that fluid goes through a large vessel called the large lymphatic vessel called the cisterna chyli. So there's a thing called the cisterna chyli right here, okay? Because it does come from your abdomen as well, so from your digestive system, and your lymphatic system picks up fats from your digestive system, that cisterna chyli can get bigger after you've had a fatty meal. So once it's absorbed more of that chyle, more of that fat, that cisterna chyli will expand. Okay? So all of that goes to the cisterna chyli, up what's called the thoracic duct, and then that drains into the left subclavian vein. And it's that simple. So your lower legs, your abdomen, and your back all drain into that cisterna chyli. That fluid's going to go up a thoracic duct and then empty into your left subclavian vein. But then also, your left arm, your left chest, and your left head and neck are all going to drain also into that thoracic duct. Not through the cisterna chyli, because that's at your abdomen. We're already past that once we get to our shoulder and our head and neck. But your head and neck and the arm and shoulder are going to go into the thoracic duct and then into that left subclavian. So the thoracic duct goes all the way up your abdomen and chest, and it curls over to the left subclavian. So your arm and your head and neck are going to drain into that thoracic duct before it goes into the left subclavian. Yeah.
And it's always going to drain into a vein, not.
Yeah. Yeah. And it's always going to be the subclavians, either the right or left. That is for the left side. For the right side, it's a little bit easier because we've already done the abdomen, we've done the back, we've done the lower legs. All of that was handled on the left side. So the right side is a much shorter pathway. It's just the right side, arm, chest, head, and neck. All of those drain into what's called the right lymphatic duct, and then that goes into the right subclavian vein. Okay, so right arm, right chest, right head and neck. Its duct is the right lymphatic duct, and then from there into the right subclavian vein. So the thoracic duct for the left side, the right lymphatic duct for the right side. The cisterna chyli, which I explained, but if you want to write it down, it's an enlarged sac of lymph and it may expand after a fatty meal. Yes. I think it does get more of a light color or a white color from the lymph. Okay, everyone good here? Okay. So you can see this here. The thoracic duct, let me make sure I find it. Here's the cisterna chyli. The thoracic duct goes all the way up and then empties into the left subclavian. The right lymphatic duct is much shorter though. Right lymphatic duct is much shorter. It's just right here. Just this small section. It's receiving lymph from something called the jugular trunk, which is coming from the head and neck, and the subclavian trunk that's coming from the arm and the shoulder. And then it drains into this really small duct called the right lymphatic duct. You don't have to know those other ones, but they're just draining separately from the head and neck and from the arm. That's why the right lymphatic duct is so short, because it receives it from two areas, and then it from the right lymphatic duct dumps that into the right subclavian. All right, so lymphangitis. Lymphangitis is inflammation of your lymphatic vessels, okay, due to an infection. So this results usually from a bad bacterial infection. And the reason why you see that redness, and what's interesting about lymphangitis, is that you can trace the infection. So you can see the red streaks, you can see how far that infection has gone up that arm, how far along that lymphatic system it has gone. Okay. That's what it's tracing there. Those are lymphatic vessels going up the arm, and they're all congested, all inflamed and infected. Yeah.
I just thought about B-ones, and that literally happened to me.
It could have been an infection from the bee itself, but yeah. Yeah. Yes, lymphatic vessels. Or it could have been the irritation from whatever was in the stinger that went up your lymphatic vessels. Why you see the redness there is because, do you guys remember what the layer of your blood vessels was called that provides blood and nutrients to the vessel itself? The vasa vasorum. So why you see that redness is due to a congested vasa vasorum. And so you get redness, and then that's also going to be, was it tender when you had it? Yeah, and there's tenderness as well with it. Treatment for this would just be wound care, obviously. That person has a wound on their hand. And also antibiotics. Now, this infection could be viral, or it could be fungal, but bacteria is most common with an infection like this. Most common is streptococcus. Strep A is usually the common cause of lymphangitis. Okay, so how lymph is transported. It's just like what we learned with veins. So lymph transport relies on something we've already described before. It's called skeletal milking, where as you move, your skeletal muscle pushes up against those vessels and forces the fluid forward. Just like veins as well, these lymphatic vessels will have valves too, so that when the skeletal muscle does push that fluid forward, a valve will catch it along the way. As it moves forward, a valve will catch it, moves forward, a valve will catch it, so you don't have backflow. And then also, just like veins, it also relies on changes, a triangle means changes in pressure at the thoracic cavity. So as you breathe in and out, and as your rib cage opens up and goes back, you're experiencing changes in pressure, which can make like a mini vacuum inside of your chest. And so that fluid in the lymphatic vessels of your chest and your abdomen can then get sucked into the subclavian veins. Just like veins. And this is all because, just like veins as well, lymphatics have a very thin, smooth muscle layer. And so they can't really rely on vasoconstriction to be enough to push the fluid forward.
Professor, you said they cannot rely on.
No, this is what they rely on. They rely on skeletal milking. So they rely on the skeletal muscle around the lymph vessels to push the fluid forward. They rely on valves to keep it moving forward so there's no backflow. And then they rely on changes in pressure to create a vacuum so that that fluid gets sucked into the subclavian veins. Okay, so lymphedema, another clinical note. This can happen due to a few different things. So lymphedema is a swelling because of excess fluid that your lymphatic system was not able to pick up. Okay. So this could be because of a blockage. Of lymphatic vessels. Could be due to an infection. One common infection that causes lymphedema is parasitic worm infections. Which white blood cell would be fighting off any parasitic worm infection? Eosinophil. This also, the blockage of a lymphatic vessel could be due to a tumor as well. So if a tumor grows where lymphatic vessels are trying to pick up fluid and they can't, then you'll have lymphedema. Also, this sometimes happens with cancer surgery. A lot with breast cancer surgery and mastectomies. So when you have a mastectomy and breast tissue is removed, usually lymphatic vessels are removed along with it because they're all attached to that tissue. And so what can happen, or what does happen with women that have mastectomies, is that they get swelling in their chests because the lymphatic capillaries that used to do that drainage have now been removed along with the breast tissue. And so they'll have to have drainage tubes, probably get lymphatic massages as well to help with the draining with whatever leftover capillaries that they do have. What's really interesting about our lymphatic system and cool about our lymphatic system is that whatever pathways are left over, those pathways can enlarge over time. So even though you have a blockage, whatever capillaries remain, those vessels can actually enlarge to help make up for that lack of drainage. So they can adapt to new situations. Yeah.
And you said treatments include like lymphatic massage.
Yeah, and drainage tubes that they'll have to have for a certain amount of time depending on the severity of their surgery. And not even just whole mastectomies, lumpectomies, things like that will also remove lymphatic capillaries along with it. Yeah.
How we do know it's a lymphatic edema or is it regular edema?
So when it's because of a blockage of a lymphatic vessel, it's lymphedema specifically. So we can have general edema. We talked about like pulmonary edema and peripheral edema due to congestive heart failure. So there are times where we just use the word edema generally, but if it's specifically because of an issue with lymphatic vessels and lymphatic capillaries not being able to pick up that fluid, then we call it lymphedema.
I have one more question.
Yeah.
In the hospitals or in clinic centers, they give out like a water pill, so they just drain out the excess.
Yeah, that can happen as well. So you can take diuretics to help take out that, but you'll still have localized swelling. So it'll help get rid of some water that could be pooled in that direction, but you're still going to have localized swelling wherever the issue is. So next we're going to go over the cells. So lymphoid cells in tissue are found in lymphoid organs, and they're found in connective tissue as well. So we talked about lymphocytes before, but now we're going to specify them. We have B lymphocytes and T lymphocytes. So our T cells, which are our T lymphocytes, they are immune messengers. When they are immune messengers, we call them helper T cells. So helper T cells help with the activation of the other T cell I'm going to talk to you about, and also B cells. And they also warn other immune cells of invaders. So sometimes they can like take a piece of a bacteria or virus and display it on their plasma membrane and say, hey, I found something that doesn't belong, somebody come get it. And they don't do really any fighting directly, they just help activate and notify the rest of the immune system. Our other T cells, they have the ability to attack invaders directly, and we'll talk more specifically on how they do it in the next lecture on the immune system, but these ones we call cytotoxic T cells because they're toxic or they kill other cells. Now, why they're called T cells, because their maturation is triggered by a certain hormone that you should already know about. Thymosin. So they are called T cells because maturation is triggered by thymosin, meaning they mature in the thymus. So that's why they're called T cells. T for thymus. Our other lymphocyte, our B cells or B lymphocytes, their job is to make antibodies. And those antibodies will help fight off foreign invaders. The specific ones that make antibodies, those are called plasma B cells. And then we have other ones that help us remember when we've been infected by a particular pathogen called memory B cells. Those are the ones that stick around for years to remember infections in case we're ever infected with that same pathogen again. And then another word for foreign proteins is an antigen. So antigen is really any protein that is recognizable by your immune system. Any protein that could be recognized by your immune system is an antigen. They can be foreign, they can be self, but any protein that can be recognized by your immune system is considered an antigen. One thing is that you cannot tell the difference between a T cell and a B cell underneath the microscope.
How do we, well, not we, but how do they differ?
I think they would probably have to get a sample and maybe, I'm not really sure how they tell the difference underneath the scope. They'd have to do further kind of testing, genetic or whatever, yeah. By your immune system, yeah. And again, antigens, we have foreign antigens that are destroyed by our immune system. We have self antigens that are ignored by our immune system. Hey, that's part of the self. I don't need to interact with that particular antigen or cell. So yeah, any protein. Could be from the body itself, could be foreign. But anything, any protein that's recognizable by your immune system is an antigen. I'll have to look that up, how they tell the difference, but I don't think they do it underneath the microscope because you can't. They look the same. And then we have phagocytic monocytes outside the blood vessel. Now, when they're in the lymphatic system, you can consider them either monocytes or macrophages. Either one's fine. Your textbook says monocyte, but whether we call them monocytes or macrophages when they're in the lymph vessels, because technically it's outside of the blood, different sources say different things. So you can use either one, monocyte or macrophage, for inside the lymph vessel. But your textbook will call them monocytes. Any questions about these cell types? So again, your lymphocytes are the main cell of our lymphatic system. So lymphocyte, lymphatic system. Monocytes are also a really big help, but they're secondary to lymphocytes. Lymphocytes are the main player for the system. And so here's a micrograph. So macrophages or monocytes and our lymphocytes are supported by something called a reticular network. And what the reticular network is, and it's also referred to as something called a stroma. And we'll have a stroma for the lymphatic system. We'll also have a stroma for the respiratory system as well. Just make sure you know the differences between the two. But this reticular network, or stroma, is a network or mesh that structurally supports other cells in lymphoid tissue. So what the stroma does, it makes all of these reticular fibers so that the lymphocytes and the monocytes have something to be anchored to. So they're just not free floating in the fluid. Okay, because they're going to much easier mount a response if they have something to structurally support them to do so. I guess black wasn't the best color to use on this, but you can see all these fibers throughout are there so that the lymphocytes and monocytes have something to be supported or anchored to. So reticular cells make the reticular fibers for the reticular network that gives them structural support. And these reticular cells, their tissue type is connective tissue proper. Do you guys remember the cell type that makes up our dense regular connective tissue? What they're called? They start with an F. Fibro something.
Yeah.
Fibroblasts. So these reticular cells, I know that was a long time ago. They are fibroblast-like. So they very much look like and act like fibroblasts.
That's what the stroma is.
Yes. Yeah. Yeah. Well, they're reticular cells, but they're very similar to fibroblasts. Just trying to get you to wake up a little bit.
I've recently reduced my caffeine intake.
You reduce your caffeine
Yeah.
significantly? That's tough. Uh, I like caffeine way too much. Anytime I try to quit, it's the worst. Headaches, nausea, it's the, oh. So I just don't quit anymore. All right, so now we're going to get into primary versus secondary lymphoid organs. The difference in definition for these is the primary lymphoid organs. This is where lymphocytes mature. Oh, and one thing I want to mention. So, where did I say our T cells mature? Thymus. B cells are called B cells because they mature in the bone, specifically in the red bone marrow. Okay, so B cells, forgot to mention this before, B cells mature in red bone marrow. B for bone. Okay. Yeah.
Because our thymus deteriorates, do we continue producing our maturing T cells throughout our entire life?
Well, once you get to a certain age, so like I'm 36, I'm not really going to be making or maturing any new T cells, not really at any high rate. So as you get older and your immune system's more and more developed, you've really been exposed to a lot of the pathogens that you're going to be exposed to in your life. The only things that are going to be different is like a new strain of the same virus. So a new strain of influenza A or a new strain of the cold virus or things like that. So you're not really making or maturing new T cells at a high rate once you get to adulthood.
Okay.
So you need it less and less, which is why it degenerates over time.
So does our immune system weaken? Or
I mean, generally, yes, as you get older, it does. I mean, like now I wouldn't say like in your 30s, or once you get older
Right.
and your older ages, once you're considered a senior, yes, your immune system will start to dwindle down. Okay, so primary lymphoid organs is where lymphocytes mature. Secondary lymphoid organs is where lymphocytes encounter antigens. So in the secondary lymphoid organs is where they're going to come into contact with potential foreign antigens that they'll have to capture and destroy. So our primary lymphoid organs where lymphocytes mature are the thymus, where the T lymphocytes mature, and our red bone marrow, where the B lymphocytes mature. And then our secondary lymphoid organs, where they're going to encounter those potentially pathogenic antigens or organisms, is our lymph nodes, is one of them that we'll talk about here in a second. Tonsils as well. The spleen, the Peyer's patches, and the appendix. Now the tonsils, Peyer's patches, and the appendix, not the spleen, not the lymph nodes, but those three, tonsils, Peyer's patches, and appendix, those three are part of something called MALT. And MALT stands for mucosa associated lymphoid, or you could say lymphatic, lymphoid tissue. So those are all lymphoid organs that are associated with mucosa. And if you remember from B32, your digestive system has that simple columnar epithelium, and there is a specific unicellular exocrine gland that's within that simple columnar epithelium. What's that called? Goblet cells. Okay, and those goblet cells make mucus. So you have a mucus lining from your oral cavity all the way down to your anus. Okay, so these three organs or sets of organs, like your tonsils, like your Peyer's patches in your intestines, like your appendix, those are all associated with that mucus layer in your digestive system. So your tonsils, Peyer's patches, and your appendix are part of MALT, mucosa-associated lymphoid tissue. which is important because you could be possibly introducing lots of different pathogens from the food that you eat through your digestive system. Yeah.
So when people get their appendix removed, is it because it was infected? Or-
Yeah. Yeah. So you get an appendicitis. So your appendix used to be really important back in the day when we didn't really have all the food preparation standards that we do now. Now it's considered a vestigial organ and we don't really need it as we did before. But what it was like a little sack of bacteria and immune cells that could help fight off an infection from badly prepared food. And so now it does have the ability to become infected. If bacteria get inside there and grow, enlarge it and inflame it, it has the ability to become infected and it can burst. And then when it bursts, then you would now have bacteria spread all throughout the inside of your abdomen and it could lead to sepsis, which is why as soon as you get that pain and they know you have an appendicitis, they want to remove that so that it does not rupture because then that could cause a really, really big problem. Yeah.
So what about when somebody has food poisoning and like being exposed to not well prepared food?
Well, then we have other treatments to be able to take care of that, antibiotics and such. And your appendix could help potentially with that. But if you get an appendicitis, it's much better to just remove it because of the potential dangers from it. Any other questions?
Did you say where the Peyer's patches came from?
Small intestine. And so these tissues, tonsils, Peyer's patches, and appendix, there's essentially a cluster of cells very similar to lymph nodes. And this is especially true for your tonsils. But their job is to first survey things that come through. If it is pathogenic or foreign, to then capture that thing and then destroy it. But their main job is to survey first. Then if they do find something that doesn't belong, that could potentially be harmful to the body, then they capture it and destroy it. Okay, questions about primary versus secondary lymphoid organs? Yeah.
Your tonsils, is that what's inflamed when you have like a sore throat?
Yeah.
It's like,
Yeah, and the tonsils that you see are your palatine tonsils, but you have 3 different sets of tonsils that we'll learn about today. You have the palatine tonsils, you have your pharyngeal tonsils at the very top, and then, oh my God, what's the third one? Pharyngeal, and then sublingual tonsils underneath the tongue.
So those 3 jobs there belong to what?
Your molt, so the appendix, Peyer's patches, and the tonsils. But your lymph nodes will do the same thing. So I guess you can add lymph nodes to that too. Okay, so now we're going to get into lymph nodes. And your lymph nodes are going to filter lymph and house our lymphocytes. Okay, so regional lymph nodes. So you have 3 different regions where you have a high concentration of these lymph nodes. So you have cervical nodes in the neck, cervical lymph nodes. You have axillary lymph nodes in the armpit. And then you have inguinal lymph nodes in the groin area. So make sure you know those 3 regions. Cervical lymph nodes, axillary lymph nodes, inguinal lymph nodes. There's a question on the exam, and I don't know if it's the second exam or the final that everyone always gets confused on, but it points to one of those 3, and it says to give the term for these structures and give both the location and the name of the structure, and that's what we're referring to here. Is it the cervical lymph nodes? Is it the axillary lymph nodes? Is it the inguinal lymph nodes?
Term location or what?
The location and the name of the structure. So lymph node function, make sure you know it's to survey, right? The main job of our lymphocytes is surveillance. And so one thing to know is that lymph nodes are our most important secondary lymphoid organ. This is where pathogens are most likely to be caught and destroyed, is at the lymph nodes. Yes, it happens in malt and the other structures, so at the tonsils, at the Peyer's patches, at the appendix, but most of it is going to happen here at the lymph nodes.
It's the site where the most captures.
Capturing and destroying of pathogens is going to occur, yeah. All right, so one thing to know, the lymph node does have an outer capsule, which is going to protect the lymph node itself, as well as the rest of the body from the potential pathogens that are inside. So there's a capsule on the outside. And then we have, just like we have for many other organs, we have a medulla and we have a cortex. So an inner region and an outer region. I used to go over the ins and outs of the differences between the two, but we don't really test on it, and I don't really think it's all that important. And I always thought it was really mean to go over those details and then never ask you about it, ever. So just know the general structure. We have a capsule for protection. We have a medulla and we have a cortex. Both the medulla and the cortex, so everywhere within the lymph node, is where the lymphocytes reside. So they live throughout the lymph node. That's all I'm going to say. They are like a bean shape. They're only about an inch in size, 2.5 centimeters. So not very big at all. What you really should know about, and what I will ask you about, is the function. So there is an afferent side that fluid will come into the lymph node, and an efferent side where fluid leaves. Okay, so afferent coming in, efferent going out. And so, hold on, let me make sure I didn't miss anything on your notes. Yeah, okay. We're still good. Yes. Okay. So afferent side for the fluid coming in, efferent side for the fluid going out. What do you see as the main difference on this picture? The difference between the afferent side and the efferent side.
There's a lot more efferent.
Yeah, so there's more afferent vessels coming in than efferent going out. Okay, so more afferent than efferent. So think of it this way: if we had more water coming into a pool than we had hoses to drain the fluid out, what would happen to the water in that pool? It would overflow, right? So what this does, having more afferent, more fluid coming in than fluid leaving, it allows that lymph node to fill up and it slows down that flow. Okay, we have lots coming in, only a couple of vessels leading it out. So it really slows down flow of that lymph fluid, which allows those lymphocytes to have a lot of time to survey that fluid. If we slow down the flow, then they can really take their time in making sure and comb through that fluid to make sure that there's no foreign antigens there. Okay, does that make sense?
Is there a reason that it's going so?
Yeah. So the reason why we have more afferent vessels bringing fluid in, then we have efferent vessels draining the fluid out, is to slow down the flow of the lymph to give the lymphocytes more time to survey the fluid. If they have more time to survey, then they're more likely going to catch those things that don't belong. So it slows flow. It's kind of like sand through an hourglass. It's really, really slows down that flow. Allows lymphocytes and macrophages or monocytes to complete their function. So to fully survey that fluid. You can say immune function. Okay, so a clinical note on lymph nodes is something called buboes. So buboes is when the lymph nodes become inflamed. And it could be due to infection. It could be also due to cancer. So you can see how this person here, his inguinal lymph nodes are very much inflamed. You can even see the axillary and the cervical ones inflamed as well. And so the buboes that you see here are from Hodgkin's lymphoma. Note the swelling in the cervical, axillary, and inguinal lymph nodes areas. So what lymph nodes can act as, they can act as a secondary cancer site. So sometimes cancer cells are able to be dislodged or escape the origin of the tumor. And so they can flow through the blood, they can get into our lymph system. And if cancer cells get into our lymph system, they can get trapped inside the lymph nodes. And then once they're trapped inside that lymph node, they can begin to grow and make more cells within that lymph node. And now you have a concentrated area of a bunch of cancerous cells. And so these buboes are usually painful with an infection. So these buboes are caused by an infection. They're usually painful or tender to the touch. But if it's buboes because of cancer, they're usually not painful. Now, a name for the condition of having these buboes, you may have heard people say that they have swollen glands. That's not particularly true because glands excrete things. Lymph nodes are not glands, they're lymph nodes. And so the name for the condition of having these buboes is called lymphadenopathy. So we will not call them swollen glands because they're not glands. So lymphadenopathy. So questions about buboes. Okay. Anybody still need these notes up? Okay. And don't freak out and jump to conclusions if you feel swollen and they don't feel painful. I mean, definitely get them checked out if you have swollen lymph nodes. But don't just jump straight to cancer. These are general understandings.
They're really bad down in the private.
Oh, they are. Yeah. All right, so lymph node sampling, why we may do a lymph node sampling is to see if cancer has spread. Rather than taking a tissue biopsy. Either way, the procedure is not all that fun, but they can take a lymph node sampling from usually axillary region or other regions as well to see if there are cancerous cells present. If there are cancerous cells present, that means that the cancer has spread and could potentially be really anywhere in the body. Because as you know, that lymph fluid goes back to your subclavian veins and then just circulates all over the body all over again. And what's the word that you learn in Bio 32 for the spreading of cancer? Metastasize, yeah. Or metastasis. All right, so the spleen is next. The spleen has a thin capsule around a bloody sponge with lots of blood going through the spleen. Remember that it is a site of hematopoiesis in a fetus and a very young infant. So as the fetus or infant's red bone marrow is developing and they're not able to make as much red blood cells to supply their body enough, then their liver and spleen can also do the blood cell production as well until their red bone marrow is developed enough. So it's a site of hematopoiesis in the fetus. Macrophages filter out old red blood cells and recycle iron to blood for liver. We talked about the whole red blood cell recycling process, how it breaks up the heme group and the globin group. The heme group is broken down to the iron and the bilirubin. The bilirubin gets converted into bile by the liver, all of that. So it all starts at the spleen though. That red blood recycling starts at the spleen. Also, it is a extra supply of platelets and white blood cells if we need it. So it's like a little immune cell reservoir that we have. So if we have a bad infection, our spleen can then release some white blood cells to help with that particular infection. Anybody remember how we store iron, what the form of iron we store it in is called? Yes. That makes me so happy. Okay. So extra supply of platelets and white blood cells if needed. Mostly it's going to be monocytes. But monocytes are so helpful. They're very nonspecific, so they can really fight off anything. And then splenectomy is a surgical removal of the spleen. However, if you do remove the spleen, now you have a lowered immunity because you don't have that extra set of white blood cells. Okay, you don't have that extra. It's a blood reservoir as well, so it has lots of blood within the spleen. So you lose that amount of blood that came from the spleen. You lose that little bit of white blood cells that the spleen holds on to for you. And so by removing the spleen, you lose those functions. You lose that little bit of a backup of blood and immune cells. So that's that. And we'll talk a little bit more about what happens when you remove the spleen in the next slide. So any questions on this, about general things about the spleen? Okay, anybody still need this stuff up? Okay, so a ruptured spleen. So like we mentioned in the previous slide, the spleen has a thin capsule. And let me make sure I'm not rewriting too many of the notes that are already on your lecture notes. Get it out. So like we mentioned in the previous slide, that there is the spleen has a thin capsule. And so it may rupture due to trauma or a direct blow. Okay. Or it could rupture from a severe infection as well. So it can rupture due to a direct blow or direct hit to the spleen, or severe infection. But either way, this is going to lead to internal bleeding. So it'll lead to bleeding in the peritoneum. Okay, and for the, let me see what I want to say. Symptoms of a ruptured spleen. Oh, and the direct hit should be in the upper left abdomen where that spleen is. But the symptoms are going to include abdominal pain, and tenderness. And I think it's already in your notes. And then left shoulder pain. Left shoulder pain is something we refer to as Kerr's sign. And so what is that word? And we brought it up before, where you have an internal issue and the pain shows up on some other external site on the body. Referred pain. So Kerr's sign is a form of referred pain where you have bleeding in the peritoneum, but it causes left shoulder pain. And right at the tip of the left shoulder is Kerr's sign. Okay. So anytime you have blood in the abdomen, you should have that Kerr's sign, that left shoulder tip pain. So this could be due to a ruptured spleen. It could be due to an ectopic pregnancy. It could be due to a ruptured appendix. Okay, so anytime you have bleeding that's occurring in the abdomen, you can have Kerr's sign. And then you'll also have symptoms that are associated with hypovolemic shock. Because again, your spleen is a large reservoir of blood. There's lots of blood there. And you have a ruptured spleen, you lose a lot of blood there, then you'll show signs of hypovolemic shock. So the thing with a ruptured spleen about why you may or may not want to remove it is that the spleen can actually repair itself. It can fully regenerate in children that are less than 12 years old. And even as adults, it can repair itself. So they are doing splenectomies less and less often now because of its self-reparative functions. And the fact that if you remove it completely, you lose that immunity and you lose that bit of blood that the spleen holds for you. So because it has the ability to repair itself, that's no longer like the first mode of treatment when you have a ruptured spleen. They're not going to immediately remove it. It's going to depend on the amount of damage that's been done. But it can repair itself. But if we do remove it, any functions that the spleen had, like the red blood recycling and such, the liver and the red bone marrow will take over the functions of the spleen when removed. Yeah?
I know how you said it regenerates in people less than 12 years old, so if we're older, would it be like the liver where it doesn't fully regenerate?
Yeah, it's not going to fully regenerate, as you get older. But it can still repair itself, but it's not going to have like you're getting a full regeneration like it would with a child. Children have much more stem cells than we do as adults, so they have the ability to regenerate a lot better than adults do. Okay, questions about ruptured spleen? Okay. So the MALT, again, we already referred to the MALT, mucosa-associated lymphoid tissue. It guards the body's entryways against pathogens. So these things are going to be lymphoid organs that are associated with the mucosa of your digestive system. So that includes our tonsils, our Peyer's patches, and then our appendix. Our Peyer's patches are very similar to tonsils, and our tonsils are very similar to lymph nodes. So they're all very similar to each other. And then we already spoke about appendicitis. So if the appendix were to get infected, it can cause an appendicitis, which is very dangerous because if it does rupture, then you can spread bacteria all in the peritoneum or the abdomen. You can also have inflammation of your tonsils, called tonsillitis. If you have them often enough, your doctor may suggest a tonsillectomy, a removal of your tonsils, and possibly your adenoids. And your adenoids are your pharyngeal tonsils, which are at the top of your nasal cavity. I've actually had my tonsils removed. I had like so many ear infections and colds and flus growing up. They removed it. Removed my daughter's too when she was only a year and a half. Oh, it's very painful. Yeah. Oh, it's so sad when you have a baby that goes through surgery and then they wake up from the anesthesia and they're just like, they don't know what's happening. They're so scared. Oh my God, it was the worst feeling, the worst experience ever.
You said the adenoids are about a year?
No, they're up at the top of the nasal cavity. So pharyngeal tonsils are what they call adenoids. So they're at the top of the nasal cavity. Then we also have our palatine tonsils at the back of the throat, which is the ones that most people refer to when they have tonsillitis, are the ones in the back of the throat. And then we have lingual tonsils at the back of the tongue. So these are very similar. Tonsils are very similar to lymph nodes, except there's not a capsule. So tonsils just don't have a capsule like the lymph nodes do. We're almost done. We're going to get done on time. I love when the lectures don't go over. But the thymus, or T lymphocytes, mature in the thymus. The thymus secretes the hormone thymosin, which activates new T cells, which you should already be familiar with. When we talk about they activate new T cells, they make them what we call immunocompetent. And all that means is, and we'll talk about it more with the immune system, but immunocompetent means that the immune cell is able to tell a self cell from a foreign cell. They know how to tell the difference between something that is potentially harmful to the body versus something that's not harmful to the body. That's being immunocompetent. Okay? They know which things to look out for. They're properly trained, and we'll talk about the trainings that the T cells go through in order to become immunocompetent. T cells can replicate once they're already made, which is why adults don't need the thymus like kids do. So once we activate a particular T cell, we don't need the thymus anymore to make copies. The T cell can make copies of itself. What it does is that the T cells, when it needs to make more copies, will release an interleukin that binds to itself and allows it to make more copies. What kind of signaling is that called? Autocrine. So it has the ability to do autocrine signaling to make more copies of itself in times of an infection. Oh, before we get there, there is something called the blood-thymus barrier. You've heard of the blood-brain barrier before because our brain is very sensitive to chemical changes, so we need extra layers to protect the brain from the blood. We also have a blood-thymus barrier. And this keeps immature T cells away from the blood. Because you do not want a T cell that's not fully trained to be in circulation. Because then they don't know the difference between a self cell and a foreign cell. And they may start to attack our self cells, which is why we have autoimmune disorders in which T cells are not properly trained or other immune cells are not properly trained and they begin to attack our own cells. Yeah.
Is that kind of what was happening when children with that had COVID were getting multi-system organ failure because their immune cells were attacking their healthy cells?
I believe that's what was happening, yeah. The same thing that happens like with leukemia, where cells are being released too early. They don't know the difference between self and non-self, and they begin to attack our own cells. Okay, last thing on HIV, which is human immunodeficiency virus. HIV replicates inside of our helper T cells. And so what happens with it and what happens with HIV and why HIV is so hard to treat is because HIV doesn't replicate very well. Okay? It has many errors when it replicates. And so it has a high mutation rate. And so it develops resistance to medications really fast. Because if it's making many errors, the HIV that you had 1 month may not be the same HIV that you have 3 months later. It may be structurally a little bit different because it makes so many errors when it copies itself. And so sometimes when it makes those errors, it makes itself a little bit different than the previous version that it was. Now the medication that you're taking is no longer effective against this new HIV that looks a little bit different than when it started. It just really is not good at making copies of itself. It tries to do it as fast as it can and just makes many errors. And so then proteins can change shape, which then can make it hard for medications to bind to it and things like that. It destroys the helper T cells when it uses them to replicate. And so then B cells and cytotoxic T cells don't get the help to fight an infection if our helper T cells are being killed. Because helper T cells are there to, one, tell the other cells when something is present that shouldn't be, and also helps activate those other cells. And so if we don't have helper T cells, then we can't activate our other immune cells. And so they're no use to us if they can't be activated. So one thing that we do in order to try to study HIV and its effects is we actually will generate or breed athymic mice. So we'll breed mice that don't have a thymus and therefore don't have T cells. And so then we infect them with HIV. I know it may not seem ethical to a lot of people, but we infect them with HIV, and then they will display symptoms that are similar to people with HIV that don't have T cells because the HIV has destroyed their T cells. So we breed mice without a thymus to kind of see what the effects of different medications are. And so HIV can develop into something called acquired immune deficiency disorder or syndrome, deficiency syndrome or AIDS. And make sure that you know that HIV can be spread through blood, semen, vaginal secretions, and so it's spread via needles, sex, and childbirth. And this is how you contract HIV. You cannot contract AIDS. HIV develops into the syndrome of having a deficient immune system. All right, any questions on the lymphatic system? Perfect timing, 9:24. Do not forget about that respiratory PLA. It's not due until Wednesday, but you don't want to wait until the last minute. It's lots of terms, lots of definitions, and you really want to get that done before we talk about the physiology, or else some of it might not make sense. So make sure that you start working on that this weekend. I have a couple of copies with me now, but only about 5 if you want to grab one, and then I'll have more copies in lab.